There are 86 billion neurons in the brain, and all of these neurons work together to carry out complex computations so that we can walk, talk, and remember. And for the brain to carry out all of these important functions, they need a lot of power. In fact, while the brain occupies only 2% of the volume of the human body, because it carries out all of these important functions that need energy, it ends up using 20% of the total energy of the human body. And when the brain does not receive this energy, it results in many neurological disorders like ALS, very well associated with Lou Gehrig, who suffered from this disease, a famous baseball player. Stephen Hawking was also affected by a form of ALS. It results in Parkinson's, as we know, a famous personality, Michael J. Fox, who suffers from it. And it also results in Alzheimer's, which results in loss of memories. This innate vulnerability of the brain to depend so much on energy that even small limitations in energy supply results in these disorders is partly due to the innate shape of the fundamental cells of the brain, which are the neurons. If you look at a neuron, you will see that it is very different from every other cell in the human body. If you take a skin cell, a muscle cell, or a white blood cell, they are all round. And they're about the size of the cell body of a neuron, which is also around, it's about round, and it's about 20 micron in size. To give you a spatial scale, one human hair thickness is 100 microns, so it's 5-fold smaller than that. And so all the cells in the human body, except for the neuron, is just this size. But then the neuron doesn't just stay within that size, it projects all these beautiful projections that come out, called dendrites and axons. And especially these axons can project for much longer distances. They can go up to millimeters and even meters along the human body. And then the main job of these two different projections is the dendrites receive information from adjacent neurons, and then the axons transmit the information and send that information to adjacent other neurons. So if you look at two neurons talking to each other, it would look like this. So you have this axon from one neuron connecting to the dendrite, the spine of this neuron, forming this junction called the synapse. And at this synapse, that the chemical molecules from one neuron is released onto the spine of the adjacent neuron, where the spine responds to this information, and this incoming information needs to be stored. And in the process of storing it, the spine undergoes this large increase in size. And this increase in size is very important for the spine to store the incoming information and create new memories. But creating this large increase in size and storing information is not a one-person job. It is carried out by thousands of molecules that are doing a lot of different biological reactions to make that happen. For instance, this particular molecule inside the synapse, its job is to maintain the amount of calcium within the synapse. And in that process, it consumes energy. And this particular protein within the synapse, its job is to maintain the amount of potassium and sodium within the synapse. And in that process, it also consumes energy. And this particular protein, its job is to make new proteins. As you know, proteins are the fundamental building blocks of the human body, and not just the human body, they're also fundamental building blocks of synapses and memories. And this particular molecule's job is to carry all these proteins from one end of the synapse to the other so that these proteins reach the right locations. So all of these molecules, I'm just highlighting 4, but there are thousands of such molecules within the synapse that need to work, do its job, so that the synapse, when it receives information, can become large in size and store that incoming information and form new memories. So because all of these molecules are basically machines and they require fuel, they consume a lot of energy in that process. And this is one of the main reasons why the brain ends up consuming most of the energy in the human body. So where does this energy come from? We have mitochondria, which are organelles that are otherwise called as energy factories of the cells. They take this molecule, a sugar molecule in the form of glucose, and they convert it into energy in the form of a chemical molecule called ATP. And this biological reaction where they convert sugar into ATP happens within these inner structures of mitochondria called cristae. And within this cristae are another bunch of molecular machines, but in this case, they are not consuming energy, but they're actually producing energy. And these molecules, again, are lined up next to each other so that they can produce this energy. So where are these mitochondria within neurons? So if you take a green protein, or take a mitochondria and tag it with the green protein, and you will notice that within this neuron that I showed you, these mitochondria are distributed everywhere. They are in the cell body. They form these long thread-like structures in the dendrites. They form these short sphere-like structures in the axons. And if you want to study them in detail, we can use a special technique which is almost like how the spotlight is flashing on me, where you can see me better and focus on just me and not the other things in the room. Just like that, you can flash light on just one region of the dendrite and only watch what happens to the mitochondria in that region. And what we learned is that this mitochondria stayed in the same position for up to 2 hours. And that was fascinating to us. We were wondering, what are these mitochondria doing here? Why are they staying there for the whole 2 hours? So to get to why, I am going to give you an example. How many of you in this audience uses Amazon for package deliveries? Can you have hands? Okay. And how many of you actually use Amazon Prime? Okay, quite a few. Great. So you already know what is the advantage of Amazon Prime versus Amazon, right? If you have Amazon, you order for a package, it might arrive in a week or so. But if you have Amazon Prime, you order for a package, it'll arrive within a day, or maybe 2. So recently, I ran out of batteries, and I went to Amazon Prime and ordered batteries. And I was surprised that it gave me this new option, and it asked, do you want your batteries delivered today within 2 to 6 p.m.? And I was surprised. And I started thinking, the headquarters of Amazon is in Seattle. And I'm sitting here in Jupiter, and I want batteries. So did they just invent some sort of a supersonic plane to bring over all these batteries within few hours to Jupiter from Seattle? No. Amazon figured it out. It partnered with the local warehouses. And so my batteries will come from within Jupiter area, and it will come at the speed I want it. And that's exactly what the neurons are doing. So when the consumers, the synapses here, need energy, they don't have to wait for the headquarters, the cell body, to send all over the neuron to give it its energy, right? It has basically placed these local warehouses, the biological batteries, mitochondria, right next to where they are needed. And this is fascinating. And guess what? The neurons figured it out way before, and Amazon only figured it out recently, right? So this is exactly what we study. We try to understand how these mitochondria are placed near the synapses, what are they actually doing to support and provide the energy required for the synapses to form new memories. So I'm going to take you through some of the projects that are going on in the lab, what are the approaches we are taking, and also highlight our lab members who have made valuable contributions to these projects. So the first question that I want to address is try to understand what is the signal the mitochondria is receiving that tells the mitochondria that now we need to start responding, there is an energy demand coming. So to address that question, we took advantage of jellyfish. Jellyfish, as you know, are fluorescent, and they're fluorescent because they make the special protein that makes them fluorescent. And we can take that protein and we can modify it so that only in the presence of calcium, the protein will become fluorescent. So now with this modified protein, you can put that inside these synapses. So here, this synapse that I'm showing you is pretty much similar to this cartoon here, right? And then you want a system where you can release this chemical locally and then ask how this spine responds to it. And that's exactly what we do here. We, using light, use a special way to release chemicals, neurotransmitters locally and ask how this spine responds. And what we actually see is that the calcium signal increases, which means that there is signal coming into the spine when the synapse is receiving information. And now we can take the same protein and put it inside mitochondria right at the base of the spine. And what we see is that now the calcium is entering the mitochondria, which means that mitochondria, the calcium is the signal that the synapse is sending into the mitochondria saying, hey, there is information coming, you better get prepared, we need to start producing energy. And this project is led by a postdoctoral fellow in the lab, Rolene, and she also was assisted by this wonderful Benjamin School, a high school student who visited us during his summer breaks and used his coding skills to help Rolene analyze some of her data. And he's right now pursuing his bachelor's in engineering in University of Michigan, Ann Arbor. So mitochondria are receiving the signal, and how do they respond? As I mentioned earlier, to understand how mitochondria respond, we need to look at the inner structures, these orange structures or cristae, where the mitochondria have these molecular machines that produce energy. So to look at these cristae structures, we cannot use any normal microscopy that I showed you so far. You need special microscopy called electron microscopy. And so for this special machine that we need for this particular experiment, we collaborate with the electron microscopy core at the Max Planck Florida Institute and Thermo Fisher. And this project is led by a talented graduate student who in fact received a Zayas Poster Award for this work. And what he found is that when this calcium signal comes into the mitochondria, these mitochondria undergo dramatic changes in its inner structure. It's almost like it's making space for more such molecules to be accommodated so that it can expand its energy warehouse, so that it can start producing more energy than it was producing before. So is it actually ending up producing more energy? So to address that question, two postdocs in my lab, Ilika and Rolene, took advantage of fireflies. So fireflies make this protein that uses the energy molecule, ATP, and converts them into light. It's the same biological reaction that allows us to see fireflies flashing in the dark woods. So we use the technology that fireflies have developed. We take that protein from them, and we put them inside mitochondria. And lo and behold, we see that these mitochondria are indeed producing ATP in response to the signal and this dramatic change in the cristae structure that they are undergoing. But my postdoc Ilika was not satisfied with this experiment. She wanted to know what is going on. So you see a signal come in and then it disappears. So she wanted to know, where is the signal going? Where is this energy being distributed to? So she took the same reporter and she put it inside the spines. And in response to spine stimulation, she sees energy being distributed into the spines. A signal goes up inside these spines, suggesting that the locally generated mitochondrial ATP is distributed back to the spines. It's just like how I ordered batteries sitting in Jupiter. Somebody got the information, they processed it, and brought that battery back to my house. Just like that, these synapses say, we need energy. They send that information to the mitochondria. They produce the energy, and they send the energy back to these synapses where they are needed. But there is a problem here, if you think about it. So for this whole thing to happen, this mitochondria needs to stay in the same position until the whole generation distribution takes place. If this mitochondria moves away anytime during this whole process, the energy will not be distributed to the right synapse. So we were interested in understanding what keeps these mitochondria in the same place. So to address that question, we took advantage of soybeans. Why stick to only animal kingdom, right? We can also turn to plant kingdom. So we turned to soybeans. They produce this enzyme, and that allows us to label all the proteins on the outer shell of the mitochondria. And that allows us to ask which of those proteins are acting as a glue to keep the mitochondria in place. So we screened about hundreds of proteins, and we found that this one particular protein called VAP, when we remove that protein, the mitochondria do not stay in the same place anymore. So this VAP protein seems to function as a glue in keeping the mitochondria in place. Not only is it important to keep the mitochondria in place to produce energy for this synapse, but keeping the mitochondria in place also is important to produce and provide energy for a neighborhood of synapses. Just like having a local warehouse does not help only me getting batteries or my parcels on time, but it also helps within the Jupiter and Palm Beach County to receive all their parcels on time. So this VAP protein is keeping the mitochondria in place so that a neighborhood of synapses can receive the energy as and when they need it, so that they can use that energy to fuel memory formation. So in the absence of VAP, the mitochondria do not stay in place. They do not produce the energy that they are supposed to. They do not distribute them to the synapses that need them. And so the adjacent synapses also do not receive the energy. So basically, the synapses cannot work. All the molecular machines inside these synapses I showed you do not get the fuel to do their job. So they do not work, and so memories do not form. And this fascinating discovery was made by 3 students in my lab, led by a graduate student, Ojasi. She just published this research work as her first author paper in Nature Communications. The second student, Tejas, was an undergraduate from FAU, and he received an outstanding thesis award for this work and is currently pursuing his MD at Dartmouth. And the third student was a master's student, and she also received an outstanding thesis award for this work, and she is pursuing her PhD at Johns Hopkins. So why does all of this matter? This VAP protein, when it has a mutation, it actually results in ALS, the same motor neuron disease that affected Lou Gehrig. So far, nobody looked at ALS as a problem of not enough energy supply for synapses, not enough mitochondria near the synapses, and the mitochondria are not present for long enough to provide energy. And this is for the first time we are looking at ALS from a completely different angle. And so such fundamental discoveries allows us to look at these old problems from new angles and hopefully will help us tackle such diseases. So as a final note, I want to leave you with 4 pointers as to why what we do matters and why support us. As I mentioned, driving fundamental discoveries is very important to understand, first of all, how our human body functions, and when it breaks down, how to fix it. And fundamental discoveries opens up new ways of looking at how biology works, and therefore helps us develop new therapies. And the second point is, through this process, we create not only independent scientists, but also independent thinkers and future leaders. We look at this as a great opportunity to train all these young minds who will go on and have a positive influence in this world. And to get to the third point, I want to tell you a little bit about myself. I come from a big city in India, South India, called Chennai. And Chennai is very similar to Jupiter, actually. It's on the east coast of India, similar to Jupiter, east coast. The humidity is exactly the same. Believe it or not, it was one of my selling points for coming here. And sure, it has about 10 times more people than in Jupiter, but it's sort of the same. And I grew up in a very simple family of 5, 3 siblings, 2-bedroom apartment. And for us, education played a very important role. So much that during important exams, each one needed their own room. And with my 2 siblings, the 2 bedrooms were taken. So I would lock myself up in the kitchen, made my own study room, and I would prepare for my exams in the kitchen. This is actually the kitchen door you can see behind my mom. So we worked really hard. But many times, working hard alone is not enough. You need opportunities. At every stage of my education or my career, somebody saw something in me and gave me an opportunity. Be it a first internship in a neuroscience lab or getting this job here at Max Planck Florida, somebody gave me that opportunity. And that's exactly what we want to do for the next generation. We want to provide opportunities, not only for our existing scientists, so that they can go and learn new skills and new ways of looking at the world, but also to new trainees, new young minds to come in and learn all these amazing things and become future leaders. And lastly, the 4th point, I want to mention that I'm also a mom of 2 small children. My son is of the same age as my lab. Both of them came into action in 2020. And my daughter was born exactly around this time last year. And it's not easy to, take care of 2 children and also run a lab and train all these young scientists and make sure they are successful. It is not easy, but it is possible, provided you have the right support system. Special shout out to my husband, who's actually watching the kids as I speak here tonight. So yes, it's not easy, but it is possible. And with your support, when you support us, I get to tell others that if I can do it, you can do it too. Whatever the challenge that you're going through, it is possible with the right support system. So with your support, you will help us inspire the next generation. Thank you.